showing the certain mechanisms which help promotion or progression of the disease. One of those, and these mechanisms are listed here. At the end of the lecture, the students will be able to briefly discuss the Warburg effect, evasion of programmed cell death, that is apoptosis. Evasion means skipping, how they skip apoptosis. As you know that apoptosis is a programmed cell death. So definitely they skip, that's why they survive, right? And then they acquire certain properties like stem cell-like properties of properties which help them differentiate, multiply, divide, and grow. Angiogenesis, that is the formation of blood vessels. Why do they need blood vessels? Because of for the supply of nutrients and oxygen. Otherwise, they will die. And then there is evasion of host defense. That the host defense meaning that the nature has provided us with a very strong immune system which detects the foreign particle or any dangerous element which is risky to the human body, but the presence of tumor behaves differently. The body at times does not even detect because they skip the host defense mechanisms. How, we will discuss today. And then there is genomic instability, and then cancer enabling inflammation. We know that inflammation there are two types of inflammation. One which is dealing with the generalized inflammation, and the other is cancer-specific inflammation. We will see how this inflammation helps the tumor to grow. And then there is a multi-step carcinogenesis where there are acquisition of multiple genetic abnormalities within one tumor. So we will discuss each one of them turn by turn. The first one is growth promoting metabolic alteration. Now, this is known as Warburg effect. Now, we have to be very clear to understand that tumor cells or the cancerous cells, they need to have a very fast and accurate metabolism because they need the nutrients, the supply of oxygen all the time, and these provide the basis of their growth. If there is not enough nutrition in the body or the cells because and enough oxygen, the tumor cells will not be able to survive. So how do they do it? Let's see. This Warburg effect means basically the aerobic glycolysis. Tumor cells, differentiated cells is the normal tissue cells, right? They either go for aerobic glycolysis or anaerobic glycolysis. Aerobic glycolysis ends up in the end product is pyruvate, which is then further utilized by Krebs cycle. The anaerobic glycolysis ends up in the lactate. Now, what do tumor cells do? Which metabolism they prefer? Obviously, since they are very fast and active, they need a very fast pathway to survive. So they choose, or they specifically remain within the confines of aerobic glycolysis because it gives more ATP at the end. So they actually choose this pathway of aerobic glycolysis, which ends up in pyruvate, and then this is taken up by the Krebs cycle, the end product for further metabolism. So this effect of being specific with this aerobic glycolysis is known as Warburg effect. This helps them to multiply and grow in a fast manner. So the Warburg effect is aerobic glycolysis because it produces more ATP, and it is a basis by which on the basis of which they have developed this technique of diagnosing tumor cells as to how much the tumor cells are active, and this scan is known as PET scan, and PET stands for positron emission tomography, and this shows that whenever this scan is suggested or advised to a patient, it is to detect how metabolically active the tumor cells are. Now, this is the Warburg diagrammatic representation of Warburg effect. This is the quiescent cell, and this is the growing cell. That is, it could be dividing cells, normally dividing cells, or the tumor cells. So, in a quiescent cells is the glucose transport system, which actually inside the cell, the glucose is taken up, and then it is through the Krebs cycle, the ATP is formed, and the byproduct is carbon dioxide. And in certain cases, the lipids are also utilized, amino acid metabolism. They all end up in this cycle. Whenever there is deficiency of nutrients, autophagy occurs. What is autophagy? Autophagy is eating up of your own organelles, the cell's own organelles. What happens is that when a person is starving, continuously starving, and during starvation there is not enough nutrition for the cell to take up the metabolism. What does it do? It starts eating its own constituents. The cell starts eating its own constituent to provide the metabolites to keep that metabolic cycle going on. So this process is known as autophagy. Now let's see what happens in a dividing cell or a tumor cell. There is a glucose transport mechanism, yes, just like the quiescent cell, but there is also a growth factor and ligand or growth factor receptor connection, which activates this PI3K-AKT pathway, which we went through in the last lecture also. And this AKT pathway then activates the RAS and RAF pathway. So these are the important pathways which actually causes this. They are involved in the protein synthesis, the nucleotide synthesis via the glucose. The nucleotide and amino acid synthesis happens, and there is lipogenesis via this mitochondrial metabolite. So basically, all of this production takes part in growth, and we know that growth in a tumor cells means multiplication, advancement of the tumor grade and a stage at times because of the because it has a fast spread rate also. And then there is this gene which is known as MYC gene. MYC is an intranuclear gene which has the control over these over this whole mechanism or the metabolic pathway. So the conclusion is this, that the tumor cells divide fast, and all the block system like P10, which is blocking the PI3K-AKT pathway, the blocks are removed, and they take up a lot of glucose as well as by autophagy they use the metabolites provided within the cell, and they develop or they multiply. They produce a lot of ATP to get energy, to produce protein, nucleotides, amino acids, and all these products. So a quiescent cell rely mainly on Krebs cycle for ATP production. If a starved autophagy, that is self-eating, is induced to provide a source of fuel, and when stimulated by growth factors, as I showed you in the diagram, normal cells markedly upregulate glucose and glutamine uptake and provides the carbon sources for synthesis of nucleotides, proteins, and lipids. What happens in cancers? In cancers, the oncogenic mutations involving growth factor signaling pathways and MYC deregulate these metabolic pathways, an alternation known as Warburg effect. So basically, these pathways are deregulated. That is, there is no control or check and balance on these pathways, and the division or these growth factor and growth factor signaling is unlimited. So the metabolism and the division of the cell is unlimited also, and so the growth is very fast. Many tumor suppressors have activities that oppose we know that tumor suppressors, they are the brakes, right? So they put on brakes to oppose this metabolic pathway or the aerobic glycolysis, which is known as Warburg effect. And what are the main tumor suppressor genes? The TP53 or P53 and RB, that is retinoblastoma gene. These are the two main tumor suppressor genes. Besides them, there are P10 and other genes also. So tumor suppressor often inhibit metabolic pathway that support the growth, right? For example, there is a brake by P10 gene on the PI3K-AKT signaling pathway. which opposes the Warburg effect. So P10, MYC, they all oppose the Warburg effect. And why do we have to oppose the Warburg effect? Just to reduce the metabolism by cancer cells so that they could not grow, as they usually do. So autophagy, I just explained to you, it is when a nutrient-starved cell, that is, there is a starvation going within a person. So the cell is starved, right? So but the cell has to sustain. It has to have its normal function and metabolism. So they actually cannibalize their own intracellular contents. Now, autophagic pathway often regulated by tumor suppressor genes. May be deranged in tumor cells, which can grow despite marginal metabolic conditions. So metabolic conditions are marginal, but since the tumor suppressor genes are mutated, there is no block on this autophagic pathway, no control. So the autophagy, that's why you have seen that the cancer patients are so skinny, because their own cells are eaten up by these tumor cells to gain the energy for survival. This is by this autophagic pathway. Now, autophagy may help tumor cell to become dormant also. Sometimes the tumor cell become dormant. When they are dormant and non-dividing, it is very difficult to treat them by chemotherapies, by radiotherapy, because chemotherapy and radiotherapy are the treatments which are effective when a cell is dividing. But when a tumor cell is not dividing and it is in dormant stage, then these therapies like chemotherapy and radiotherapy will not function properly. Now, there is another process by which they skip death, that is evasion of programmed cell death. They simply forbid to undergo apoptosis. They resist apoptosis. How do they do that? We know that there are two pathways, intrinsic mitochondrial pathway and extrinsic fast ligand pathway, right? So intrinsic pathway is by this mitochondria, right? P53 DNA damage, P53 response, and there's activation or release of this BCL2, which is an BAX-PAC, which is the apoptotic gene, and to resist it, there is anti-apoptosis gene, which is the BCL2, BCLXL, and MCL1. Now, if pro-apoptotic genes, that is BAX and BAC, are more, then cytochrome C will be released from the mitochondria, and it activates the caspase, and eventually leads to death. Similarly, this happens in the extrinsic pathway, where FAS receptors are bind with the FAS ligand, and they activate these pro-caspase or death-induced signaling, leading to death pathway. This is the normal mechanism of apoptosis, right? But what happens in tumor cell? Intrinsic pathway is most often disabled in malignancy. Intrinsic pathway is the mitochondrial pathway. It does not function anymore in malignancy. And the prototypic anti-apoptotic gene is BCL2. That is, it opposes the apoptosis even in the tumor cells. So there is the classic example of BCL2 overexpression, that is anti-apoptotic protein. BCL2 is more expressed, overexpressed in follicular B-cell lymphomas. Okay, and there is along with this BCL2 expression, there is also an important translocation in this tumor, that is chromosome 14 to 18 translocation. So all these factors combine and provide a survival platform for these tumor cells. Now, in certain cases, there are reduced levels of FAS also, and if there is FAS is reduced, then you can imagine that the apoptosis will not occur, right? So these are the pathways through which they skip apoptosis. And when they skip apoptosis, when they skip the programmed cell death, the tumor cell, they are able to survive. And if they survive, obviously we know that they will progress and the tumor will grow at a higher stage. Now, certain tumor cells, they acquire properties of stem cells. Now, what are stem cells? Stem cells are basically the cells which divide and differentiate into any kind of tissue cells, right? So, certain tumors have different kind of hormone production. Certain tumors have different kind of tissue formation. And how do they do? Because they acquire these stem cell-like properties. Now, stem cell-like properties of cancer cells are acquired they become immortal. That is there is no death to these cancer cells. Why? Because there is evasion of senescence. Now we have to understand what is senescence. They skip senescence, they skip mitotic crisis, and then they self-renew because they have stem cell-like properties, so they can renew themselves, they can multiply, they can produce the progeny, right? So these are the 3 steps. First, we will discuss what is senescence and how do they escape senescence. Now, what is a telomere? You know that there is the end. There is a chromosomal end which is known as telomere, right? Telomere is the chromosomal end position. Now, what happens is that normally in each dividing cell, whenever a cell divides, the end portion of a chromosome is lost. So the length of the chromosome is shortens with each cell division. Because the telomere, there is a centromere, which is the dividing area, and the telomere is the end point of the chromosome, right? With each division, a telomere is lost, or there's shortening of telomeres, right? Which means that these chromosomes, they actually lose their length with each cell division. Okay, and what happens is that when there is short telomeres, then there are systems, there are cellular pathways which detect that the length of the chromosome is now reduced and that it is shortened. So what do? They detect these cells as an abnormal cell, and when they think or they imagine that these cells are abnormal, they actually give signal for these cells to die, that is to undergo apoptosis. Okay, this is called senescence. Senescence means full stop to the cell division, full stop to the cell survival. There is no survival when there is senescence. Okay, so let's see. Now there is an enzyme which is known as telomerase. Now what does this telomerase do? Telomerase continuously adds up these nitrogenous bases to this chromosomal end, that is telomere. So it does not let the telomere shorten in length. Are you getting my point? What will happen when the telomere will not shorten? It will not undergo death. As we just discussed over here, that when a telomere is so much shortened that it could not survive, the cell detects that it is no longer of use and let it die. But this telomerase enzyme, it has this function of continuously adding up these bases to this end of the chromosome, that is the telomere part. So the length of the chromosome will not shorten. It means that the cell cycle will detect that this chromosome is normal. It is not shortened in length, so it will not undergo death. And if it does not undergo death, it means it will survive, that it will multiply. So tumor cells, what happens in most of the tumor cells, there is this active telomerase enzyme which leads this chromosome or makes this chromosome survive. Okay, and this is the game of survival. Tumor fights with the body cell, normal cell for survival. It defeats the body cells so that it can win this game. And this game means that when the tumor cell wins, the human cells, the body cells, the normal cells are defeated. And the tumor wins, the patient will die. So this tumor, they use this telomerase enzyme to survive. Now, escape of cells from senescence. Now, how they escape senescence, it's now very clear. You can see that when the telomerase is not active, that there is telomere shortening. This telomere shortening in the cell cycle, the P53 detects the shortened telomere and gives the cell cycle a signal that this cell has to undergo senescence. That is, there should be no further multiplication or division, and that the cell has to die, right, by apoptosis. And P53 activates apoptosis also. Okay. Now, what happens if there are certain breaks in, like there are dicentric chromosomes, there are multiple breaks in this at the metaphase and anaphase, multiple breaks in this new double-stranded breaks in a cell, right? So what happens is that there is telomerase. If there is telomerase not active, then there is further mitosis, and if there is telomerase reactivation, then there is cancer, a definite cancer formation, because telomerase will help these chromosomes not to lose their original length, and these double-stranded breaks, this abnormal chromosome will survive. And if you can imagine that if a chromosome with a double-stranded breaks, that is abnormal chromosome, if it goes into the survival, it's definitely going to cause malignancy. So normal human cell divides 60 to 76 times, and then it becomes senescent, right? But senescence due to upregulation of P53 and INK for a P16 maintaining RB protein. In malignancy, this RB-dependent G1S cell cycle checkpoint is disrupted. So there is additional replication of these abnormal chromosomes. Now, replication of somatic cells leads to shortened telomeres, as I just told you. So there are competent checkpoints in the cell which then undergo arrest and cause senescence. That is full stop to that cycle. Now, in the absence of checkpoints, there is DNA repair pathway is inappropriately activated. So the DNA repair is inappropriately activated. The chromosomes are dicentric. There may be double-stranded breaks in certain DNAs, and these DNA will undergo the cell cycle. Now, you can imagine that if a cell cycle, a damaged DNA undergoes a cell cycle, what happens? It will cause malignancy, cancer. If cell fails to re-express telomerase, it undergoes mitotic catastrophe and death. So, re-expression of telomerase escape the bridge fusion breakage cycle, survival, and tumorigenesis. Now, evasion of mitotic crisis. Telomerase actually normally expressed only in germ cell and stem cell, is not active in most somatic cells. Chromosomal telomeres progressively shorten with each division until DNA replication can no longer proceed. This is just the revision of what I explained to you in the previous slides. Shortened telomeres are interpreted by DNA repair machinery as double-strand breaks. So, double-strand breaks, that is the shortened telomeres, they cause a mitotic crisis when the cell cycle arrests via P53 and RB. So, if there is cell cycle arrest, there is a mitotic crisis. Cancer cells overcome these limitations by reactivating telomerase. So, more than 90% of human tumors show increased telomerase activity. I hope now it is very clear of how telomerase act. Certain tumor cells acquire these self-renewal or stem cell-like properties. And stem cells are immortal. We know stem cells are found in our hair, in our skin, in our epithelia, GI epithelia. So they continuously divide and replicate, right? Because they're also capable of self-renewal. Now, cancer stem cells generated by transformation of a tissue stem cell, and conventional somatic cell is transformed into a stem cell because it acquires the properties of stemness. Stemness means self-renewal. Now, these were the three steps by which a tumor cell or cancer cell acquires stem cell-like properties. Now, the next specific property of malignant cell is angiogenesis. What do you mean by angiogenesis? Formation of blood vessel. So, tumor, they need nutrients, they need waste removal. So, they induce host blood vessel growth. Alright, they induce growth of blood vessels. We call it neovascularization, and for that they release two factors: growth factors, which is an insulin-like growth factor, and PDGF, which is a platelet-derived growth factor. You've studied these in inflammation and repair. If you remember, what do? They cause growth of the microenvironment or the fibroblast and the collagen and the blood vessels. So angiogenesis, tumors or host stromal inflammatory cells, they release these angiogenic factors, And again, if there is hypoxia, it is also a driving force for angiogenesis, that is new blood vessel formation. Now, endothelial growth proteins are VEGF and basic fibroblast growth factor. So you have studied their function well in repair, in the topic of healing and repair. Same function occurs in the malignancy. Invasion of the ECM, that is extracellular matrix, initiate the metastatic cascade. What happens that the tumor cells, let's start with this diagram. What happens that this is a tumor cell which is located in a tissue, okay? Say for example, this is a primary tumor which is a squamous cell carcinoma and it is lying within the skin. Just imagine, okay? And this skin or epidermis, this is squamous cell carcinoma. It is bounded below by a basement membrane and then a dermis, okay? So what happens is that this is the basement membrane, which is the basement membrane of the skin, okay? And now since they have formed, they start causing angiogenesis or formation of new blood vessel within that tumor area, okay? Now what happens is that they actually invade this basement membrane and goes into the dermis or the deeper tissue. Along with it, the vessel grow. So there is the passage through the extracellular matrix. Dermis has, or any deeper tissue has extracellular matrix, right? So they invade this extracellular matrix and nearby in the vessel wall, they break the basement membrane of a vessel wall and they intravasate into the blood vessel. Now in the blood vessel, there are other immune system cells like lymphocytes, like neutrophils. So they form an aggregate, or they interact with these lymphoid cells and they form an aggregate with these cells. They interact with the lymphoid cells and they form along with the platelets and these lymphocytes, the tumor cells, they form an embolus. Embolus circulates within the blood vessel, right? And then at certain points, say for example, they started from the skin of leg, and then they, along with the blood vessel, they move to the bone of the arm, right? So to cover that pathway from leg to the arm, they need to travel through this blood vessel. This is the medium of travel for these tumor cells or embolus, tumor cell embolus, right? Now now at certain point, if they need to invade if they are invading that bony area within the arm, they will now adhere to the basement membrane of this extracellular matrix or the tissue where they need to invade, right? They will break the basement membrane and the new vessel formation is already present there because the tumor cells are now gaining entry into the bony tissue or any other tissue they are invading. They extravasate, they make a metastatic deposit. We call it metastasis now because they're traveling far and away from their site of origin. Okay, so now they got they deposit into the distant site. We call it as a metastatic deposit. There again, they are forming new blood vessel because they need blood vessel for their survival. They need nutrition, they need oxygen for survival, right? And this is how they grow. This is the whole mechanism of how a tumor migrates from their site of origin to a distant site. We call it metastasis. Now, tumor cells detach. First, they detach from each other. There are certain adhesiveness, laminin, fibronectin, basement, how they are attached to the cadherins. Cadherins are the proteins which make them adhere to each other. Laminin and fibronectin are the proteins or the receptors which make them attached to the basement membrane. So all of these bondings are broken, and then this is how they are detached from each other. Then there are certain proteases that are released from these tumor cells which degrade the basement membrane. They also degrade this basement membrane. And then there is metric metalloproteinases which again, degrade the basement membrane. And this is how they invade the basement membrane. Type 4 collagen is broken down and then they migrate and reach to the blood vessel. So there is ECM attachment. The invading cell must express adhesion molecules that allow interaction with the ECM. Obviously, they need certain molecules to adhere to ECM, otherwise they can't, right? So migration, cytokines, chemotactic factors, then certain degraded components of ECM, liberated stromal growth factors, they all give signals to these tumor cells to migrate. Obviously, they need signals, they need receptors, they need adhesion molecules, otherwise they cannot they cannot make attachments and they cannot move. Now, how vascular dissemination occurs? I just showed you in the diagram that there is a vascular dissemination. Homing means they form a second home, or they form a place of adherence or residing for themselves. So, tumor cells embolize in the bloodstream and adhering to the circulating leukocytes and platelets. How it is influenced? This whole mechanism is influenced by vascular and lymphatic drainage from the primary side, adhesion molecule CD44, a specific chemokine receptor CXCR4 and CCR7 for example, in the breast cancer, and then microenvironment, that is ECM of the organ or the site. We just studied all these. This is just the summarization of what we studied in the previous slides. Now, metastasis may require certain mutations to occur in the stem cells. So, there are certain microRNA which are the metastatic suppressor genes. When these microRNAs are non-functional and not working, the metastasis occurs. Now, evasion of host defense. There are evidences, and there are strong evidences that our host system or defense system usually detects these tumor cells and try to fight in the initial stages. But the tumor cells are so strong that they defeat these immune system cells and make their place in the body. How do they do it? Let's see. So basically there is an immune surveillance. That is, we have an immune system which is capable of recognizing and eliminating malignant cells. We are blessed with it, but they take up certain roles, like we'll say that they are wearing certain kind of clothes to hide themselves, wearing certain kind of masks to skip the attack by these immune cells. They are clever cells, these tumor cells. The malignant cells are so clever that they know how to skip the defense mechanism. They camouflage just what they do in the army when the army is in the defense side, and how the spies come and they camouflage themselves and skip the border defense teams and the army at the borders, and they camouflage and get entry into a country. This is the same mechanism they adapt. So reactive, what happens is that normally when there is a malignant cell proliferating inside the body, the lymph nodes undergo hyperplasia. That is, they start overworking, so that the cancer cells are stopped right there at the lymph node and they do not proceed onwards. Similarly, certain tumors are found in immunodeficient patients only, especially those patients who have a not so active immune system. They are supposed to have more chances of developing cancers. And then there are certain tumor specific T cells and antibodies which are specifically present in the body to fight against tumors. And similarly, there are tumor antigens. Now, certain tumor antigens which activate our host immune response. These could be mutated genes, these could be aberrant proteins, these could be oncogenic viruses, oncofetal antigens, these could be altered glycolipids and glycoproteins. These could be cell type specific differentiation antigens. So anti-tumor effective mechanism. How do we know that our body has anti-tumor mechanism? Cell-mediated immunity, especially cytotoxic T cell, natural killer cells, they are specifically designed to fight the malignant cells. So it's not that the tumor cells are so welcoming to our body all the time. No, it's not. But they are so clever that they skip these defense mechanisms. Now, development of variants that can they develop different forms. As I told you, variants means they change shape, they change function. they change the routes through which they attack the body. So this is how they escape. It's exactly like how the spies of other countries, change shape, change dressing, change their language to gain entry into another country, right? The tumor cell mechanism is similar. Outgrowth of antigen negative variants, so that they adopt different variants, then they cause loss of MHC expression. And if you know that MHC expression is so important to be detected by T cells, cytotoxic T cells, and if the MHC expression is lost, then how can our T cells detect these malignant cells? So they're so clever, right? And then there is activation of immune regulatory pathways, that is certain surface proteins that induce lymphocyte death. They actually, these tumor cells cause certain upregulation of certain proteins like PDL1 or PDL2, which cause lymphocyte death. So when the lymphocyte is lymphocyte dies, say for example the army which is present at the border, if that army dies, if the army people die, then it's so easy to gain access or gain entry into the country where you want to invade, right? So these are the same mechanism that they adapt. Now, coming to the genomic instability, I also explained to you in the previous lecture that genomic stability means accumulation of multiple mutations in the DNA repair proteins. And you know what are DNA repair proteins? P53 and RB are the two major DNA repair protein which actively participates in the cell cycle checkpoints. So what happens is that if there are three types of defects like mismatch repair, nucleotide excision repair, and recombination repair. For you people to not to go into the detail at this stage, I would like you to just remember the name of these repairs, because this is out of the scope for you to go into the detail. Now, you have studied inflammation, but inflammation was, what you studied was related to the inflammatory diseases, okay? You haven't studied inflammation with the context of the malignancy or neoplasia. So what happens in these chronic inflammatory processes? Cancer provokes chronic inflammation. The chronic inflammation induces growth factors. So you know that growth factor important for proliferation of these tumor cells. Their microenvironment, the fibroblast, the new vessel formation, okay? Now, they also cause a degradation of these adhesion molecules. So the adhesion is lost, then it's so easy for tumor cells to move around, right? Metastasize. Then they cause enhanced resistance to the cell death, right? So apoptosis nahi hone denge. And then induction of angiogenesis like VEGF, PDGF, okay? And then facilitating invasion matters. Exactly अभी हमने सारा कुछ मैकेनिज्म पढ़ा डिटैचमेंट का, कैसे हो रहा था, इन्वेजन हो रही थी, इन्वेजन के बाद मेटास्टेसिस हो रही थी, इसमें सारे साइटोकिन्स इन्वॉल्व थे, कीमोकाइंस इन्वॉल्व थे, रिमेंबर. एंड देन कंट्रीब्यूटिंग टू इम्यून इवेजन. अभी इम्यून इवेजन हमने बात की है कि इम्यून सिस्टम में वेट करते हैं बिकॉज मैक्रोफेज को रिस्पॉन्ड नहीं करते, बिकॉज MHC लॉस हो जाता है. सो दीज आर ऑल रिलेटेड विद इन्फ्लेमेशन. Now, cancer associated genes की dysregulation हो जाती है. That you have studied in detail in the last lecture. So which genes or dysregulation of cancer associated genes, like there could be chromosomal changes like translocation in the chromosome, deletion, gene amplification, epigenetic changes, or non-coding RNAs and cancer. Now, there is the last but not the least topic is molecular basis of multi-step carcinogenesis. This you'll be studying in detail in colon cancer when you'll be doing the small intestine, large intestine tumors. But just to give you an idea, what is a multi-step carcinogenesis? This is the diagrammatic representation of a colonic epithelium, right? Mucosa, submucosa, muscularis, and serosa, right? So why mucosa at risk? Because there is an inherited germline mutation that is first hit in this patient, and that is the gene of APC, right? This APC gene is inherited germline mutation is present in this patient, but the cancer is not phenotypic right now. The patient has not developed cancer. But this genetic mutation is present because of the inheritance. So this mucosa of this patient, this particular patient, is at risk of developing malignancy. Now what happens is this beta-catenin mutation is added. So what happens is that there is methylation abnormalities and inactivation of normal alleles. So there was one normal allele and one deranged allele. So this normal allele is now inactivated because of the second hit, and this was because of the APC beta-catenin pathway and certain environmental factors. So now this patient will develop a benign tumor or adenoma, right? Now what happens is that because of the further acquired mutation in the proto-oncogenes, for example, the KRAS gene, this patient acquire a mutation in the proto-oncogene, KRAS gene, then there are chances that this patient, when it is starting to develop an adenoma, right? And then this adenoma, because adenoma is a pre-malignant condition, it may lead to a carcinoma. So now you can see that there is a wild type TP53. Wild type TP53 is a normal TP53. But if there is an abnormal TP53, if there is wild type TP53, because we know that it is a repair gene, right? It is a DNA repair gene. It will detect that there is an adenoma formation. It will cause a senescence. Full stop to the multiplication and cell division. So adenoma will remain adenoma. It will not move forward into the carcinoma. But if in this adenoma is further requiring a TP53 or P53 mutation, then you can see that this adenoma will convert into a carcinoma. This additional mutation will be acquired, like activation of telomerase, and also it will become a colonic carcinoma. This is the basis of multi-step carcinogenesis. I know that this is a bit of the difficult type of discussion for you at your level,